Okay, in this video, we're going to consider the mechanism of bile synthesis. So what exactly is bile? Well, the major component of bile are what are called bile salts, and we're going to look at these first. And this accounts for approximately 65% of the components of bile. Phospholipids are another component, and they make up approximately 20% of the total bile. Then we have bile pigments. These are small but important, 0.3%. And then finally we have some residual proteins that end up in the bile, and that accounts for approximately 5% of the constituents of bile. So let's look at our first bile component, bile salts. I think this will be familiar to everybody. This is cholesterol. Important to note about biosynthesis is that predominantly the largest component of bile is derived from cholesterol. We'll be revisiting this when we look at lipid transport in the blood, but the biosynthesis pathway is the major process of which we are able to rid the body of excess cholesterol that is delivered to the liver. So biosynthesis is going to occur in the liver, in the hepatocytes, and the first step of biosynthesis is going to create 7-hydroxy cholesterol. Now this step is very important. It's important to remember because this will be a question on the boards. And what you need to know is the enzyme that does this. It's called 7-alpha hydroxylase. 7-alpha hydroxylase is the rate limiting step of bile synthesis. So this enzyme is going to produce on the 7 carbon of the B ring of cholesterol, we're going to add a hydroxyl group to form 7-hydroxy cholesterol. This reaction is a redox reaction. We'll be looking at more of these types of reactions, but we are going to use the reducing power of NADPH. We will also be including oxygen in this reaction. The mechanism is not important. What is important is remember that this enzyme is the rate limiting step for biosynthesis. So once we form our 7-hydroxy cholesterol, then we are going to take several steps to produce 2 forms of cholic acid. The first form is cholic acid. I won't draw the structures out for you here because it's not important. The other one is chenodeoxycholic acid. Important to remember these two, differentiate these two just by name alone, because these are the 2 backbones of what is eventually going to be the bile salts. Now, during this process of forming these, we are actually going to derive the coenzyme A derivatives of this. So we are going to have, for example, if we look at cholic acid, from the top of the D ring, we are going to have a CoA derivative. Now recall from basic biochemistry, and we'll be going over this in detail, that this bond between the carbon and sulfur is a high energy bond. That means that this bond is very labile. Both cholic acid and shenodioxycholic acid have this coenzyme A modification. What that's going to allow us to do is perform the next step in the chemistry. And here we are going to take an amino acid. I think everybody's familiar with this amino acid. This is glycine. It's the simplest amino acid. And we are going to form this linkage. So this nitrogen is going to displace the acetyl-CoA, the coenzyme A group, and we are going to covalently bond the glycine onto this carbonyl residue. This, once we do this, this is called glycocholic acid. And this is one of our two major bile salts, bile acids. Alternatively, we can take another form of small molecule, this molecule, if many of you have cats, you know that recognize taurine. Taurine is important for kitty vision. But if we take taurine also, instead of glycine, we can attack that carbonyl with this nitrogen of taurine, and we can form taurocholic acid, which is the second major salt of bile. Now, why are we doing this? The sole purpose we're doing this is simply because it's chemistry. This group alone here, if we remove the coenzyme A modification and we make a carboxylic acid, so we're just going to put an oxygen right here instead of the sulfur alone, making this a carboxylic acid, the pKa of this is approximately 5.0. So remember, the pKa is at the pH, we have half deprotonated and half protonated. So we will have half of these groups like this, plus half of these groups deprotonated. By putting on glycine, or taurine on here, we are going to alter the pKa. So the glycocholic acid, the pKa, will be approximately 3.7. With taurine, the pKa will be approximately 1.5. Now, if you recall your general chemistry, what this means is that at neutral pH, if we do not make these substitutions, at neutral pH, the majority of the species will be in the ionized or deprotonated form. With a lower pKa, that means a greater percentage of these functional groups will be deprotonated. The greater the amount of deprotonation, greater deprotonation equals greater solubility. Now, why is this so important? It is this property that is generating the function of bile. This is how bile is able to work effectively. Now, notice the cholesterol backbone here. This is a carbon, multiple carbon ring structure here. So this portion of the bile salt is very hydrophobic. The portion that is conjugated onto this, whether it be glycine or taurine, these are very hydrophilic. By having these two different chemical properties on one molecule, we create a molecule that is considered amphipathic. Amphipathic means that there are multiple functional properties. In this case, we have hydrophobic domains, we have hydrophilic domains. Now, this is exactly how the bile is going to work in the intestine to solubilize fats. And that is the primary function of bile synthesis, is to emulsify the fats so that we have better digestion. The second also important reason is that it is the major pathway that the body can get rid of excess cholesterol.